Hello, good morning, and welcome to the International Plant Breeding Seminar Series hosted here from North Carolina State University. Thank you for joining. Please let us know where you're joining from. We have the honor and the pleasure to have Dr. Patrick Cumbie, who is the Director of Plant Product Development at ArborGen. Dr. Cumbie is in charge of plant breeding, seed production, and genomics activities, both here in the US and Brazil. Prior to joining ArborGen in 2010, Dr. Cumbie worked at Weyerhaus and as a forester researcher, and also worked as part of the NC State Cooperative Tree Improvement Program. He has a PhD from North Carolina State University, and he also has an adjunct professor position here at NC State. Patrick, it's a pleasure to have you with us, and I'm going to stop sharing and give you the floor to you.
Well, good morning, good afternoon, and good evening, depending on where you are. I'll just say thank you for the opportunity to share today. Started here. All right, so today I'm going to talk about making genetic gains improvement in forest trees.
You want to share your screen, Patrick? Want to try again? I don't see it.
Uh-oh.
There you are.
All right.
Sounds good.
Hopefully sharing now.
Yes, it is.
All right.
Yes.
So yeah, talking about making gains in forest trees and focused on pine species in the southeastern U.S. today will be the context, but hopefully what I bring up translates to things of interest to you and your discipline and your species. So I'll get started here. First with a phrase called gain versus risk. And so this was something that was repeated to me many times as a student. It started when I was an undergraduate. I was able to take a class by Dr. Bruce Zobel, who was more or less the founder of tree improvement in the southern United States and conifers. He always emphasized the balance of making gains, selection differential versus the risk of a reduced genetic base. So basically working with that idea of balancing gain versus risk of the unknown and managing diversity. So this is a phrase that most tree breeders in the Southeast, because many of them have been educated through NC State University, are probably familiar with. And so that's just a flavor of how we're trained to think in forest trees. And so if you tend to be conservative, then you will lean towards mitigating that risk and you give up some gain. And if you are more aggressive, you're more willing to give up some of that diversity and go for the highest possible gain. So really, in thinking about trees, with some of our challenges, it comes down to how aggressive are you willing to be to make those gains. And so we want to make gain, but we don't want to be reckless. So part of that reason that we tend to be conservative, we tend to think about this gain versus risk, is because of the time it takes to produce a new product or a tested family that can be deployed on the landscape. And so here's just a taste of what it looks like. So loblolly pine is the species here. This is we're probably in the middle. So it might take 25 to 30 years to get a product all the way through the breeding program, through seed production, through the nursery and out to the landscape to be planted. Some species, it might be double that. It might be 50 or 60 years. And others, it might be 7 to 15 if you're in the tropics and you're working with a faster growing species. And so loblolly pine is probably in the middle and poses, but all of these timelines pose a challenge of how to keep your development going, how to balance that making that genetic gain with the risk. Just a little bit of background or context. The southern forest industry is large, is a considerable component for most of the southeastern states of the United States. So it's over 200 million acres of forested land and almost 40 million acres of plantations. So that's really the focus of our genetic improvement, are those acres of forest plantation. And it employs as an industry almost 1 million people. So it's a significant part of the economy. And so thinking about genetic improvement is part and parcel, essential to improving the industry and maintaining the market. What that looks like in our part of the world, there's always a mix of natural stands or naturally regenerated forest plantations, and so you have a mix of species, and then you have planted areas, and so these natural areas are basically a mix of, could be 5 to 15 or 20 different species, slower growing, more based on diversity and conservation, ecological value. And then you have planted areas, and you can see the rows of pine trees. And this is the area where we're, in most cases, managing for a financial return, or a specific objective. Could be timber production, fiber for pulp and paper, possibly bioenergy or carbon. For ArborGen, our focus is in 2 parts of the world. The southeastern United States is our largest area, but we are also operating in Brazil, and that market is growing. Here in the United States, our breeding programs focus on pine, although we do sell other species, sometimes as many as 50 to 100 different species. Pine, 3 species of pine are our focus for breeding. In Brazil, the largest component is eucalyptus, which is clonally produced with vegetative propagation and rooted cuttings. And we also have about 20 million or more pine seedlings produced every year. And in general, our focus is to improve the yield, the wood quality, so biomass, and then also maintain disease resistance for our species. A lot of times we have to start with some basics with our customers, our landowners, or other stakeholders, because most of the time they look at the flowering or cones produced by the tree. They look at the small seedlings in the nursery, and they don't differentiate what things are. So they all look the same, and they don't really think about genetics in forest trees the way they might think about genetic differences that they would see in agriculture. And so usually we have to remind them that there is genetic diversity, there is variation, and there are differences in the families that are produced. We do this largely through bare root seedlings. So this is a picture of a nursery. These seedlings are grown mostly in a sandy soil that can be lifted and packaged and shipped out to customers. We do produce some containerized seedlings as well, but they're basically all grown. The culture of growing these is to produce about a 20 centimeter seedling with a 4 to 6 millimeter diameter that can be packaged and shipped to the customer. So they all look the same when they come out of the nursery, but they don't all look the same once they are planted in the field and the plantation is produced. And so here on the left is an example of what you might see with wild type loblolly pine, Pinus taeta. The tree is not straight. The branching is not uniform. This is a tree that's not going to produce a very good board or solid wood product out of it because of these flaws. On the right, you have a tree that's painted and marked. This is a selected tree for the breeding program. It's a very straight stem. It's got much more uniform and controlled branching, good growth compared to its neighbors. So it's a candidate to put into the breeding program and represents a tree that's more like what we're looking for. ultimately, those differences in the field lead to what the final product is. And so when we think about our breeding programs, what our objectives are, we need to think about that. And so that end objective is important. On the left, we have trees that have some stem quality issues. Maybe they weren't growing quite as well. And that's going to go to the pulp and paper market, which is typically a lower value product here, versus the picture on the right. This is high quality saw timber, which is going to go for a much higher value. Typically, this is a smaller percentage of the wood harvested off of a plantation, and that's where genetics offers the opportunity to increase that percentage. And so you could potentially go from, say, 30 to 50% of your trees qualifying for that high value. With some of the best genetics today, you can probably get to 90% in that highest value product class. So it can be very valuable to the landowner. When we look at where tree breeding fits into overall productivity in forest plantations, this comes from a paper back in 2004, but just kind of highlights as the industry and the discipline has progressed, we've moved from natural stands to plantations, different pieces of silviculture and stand management, forest management to increase productivity, and then developing genetics. And the paper only went through 2010. You can see this square on the right that goes to 2020. We don't really know what the potential is. We continue to work with breeding and genetic improvement. We have pursued clonal technology and biotechnology approaches. None of those have really come to maturity in the market, but that potential continues, and so we may be able to make dramatic improvements there as our genetic resource continues to develop. Here's just a quick overview of snapshot of what loblolly pine breeding looks like. And so it's an ongoing cycle through the year as we follow the seasons and different activities. But we're data driven to make the selections. We put those into a seed producing area. And so here's a top grafted tree. This is actually probably 15 meters in the air on top of a mature tree, which we use to stimulate flower production. We use pollination bags, isolation bags to make those crosses. We then grow those seedlings in containers in a research nursery, and we put them out in field trials and experimental design, and we grow those to anywhere from 3 years to 6 years in loblolly pine to make measurements and assessments. The traits that we're looking at for loblolly pine, we do want yield improvement. Ultimately, the tons of biomass or wood that are produced per hectare or per acre. We have a native disease, fusiform rust, so we're looking for disease resistance from that pathogen. We have originally loblolly pine was a fairly crooked tree that didn't produce straight stems. There's been a lot of emphasis on stem quality and stem straightness, and have made significant gains there. And so that continues to be a trait of interest to make sure the stems are straight, so the final product in solid wood is also straight and has good wood properties. And then finally, forking. This is, of the four, probably gets the least emphasis. It's a lower heritability trait, but it does get emphasis in the program. And basically, that fork makes the tree non-merchantable for multiple products. It's just hard to deal with both operationally, and the wood is not good in that fork. We combine all these in a trait we call saw timber potential. We do improvement on the individual traits, but a lot of times we describe the overall performance of a family with the score or metric of saw timber potential, which combines all of these into basically looking at what percentage of the plantation or the individual trees are quality, have sufficient quality for saw timber for solid wood. We work in 5 different regions. So there are populations here to meet the characteristics needed or the traits needed for each region. Basically, moving from east to west, it gets drier. Moving from south to north, it gets colder. And so we have both of those traits, drought hardiness and cold tolerance, worked into the populations that we work with in developing new material in these regions. We also have cooperative programs that we've been members of. There are 3 university-based programs in the Southeast. So that is also part of our base. So we draw from those programs, but we're also working on elite development for the deployment population. Here's just an example of what some more basic genetics might offer. You certainly have some good trees in this plantation. If you had good survival and growth, you would have full rows here of the trees. You see some not so healthy and vigorous looking trees here in the foreground. They're not straight. It's pretty variable. So this represents what can happen if you don't invest in genetics. Here's another picture. If you don't select something with disease resistance, you get infection. Typically, that infection forms this canker or gall in the tree that's in the first 2 meters of the tree and makes it unmerchantable. On the flip side, if you select a family, you select genetics that has good performance in all of its characteristics, you have a much more uniform plantation, you have better growth and quality, and that's going to lead to that higher value market. There are just a couple of pictures of what those good trees look like, where basically almost every tree, nearly 100% of the stems, are going to qualify for the higher value product classes for the landowner. So we see those differences in genetics, and back to that timeline of just thinking about, how do we manage, how aggressive can we be to capture gain? We look at what it takes to develop it. So just thinking about, if we start with a new untested selection or a new population of trees in 2024, it's basically going to take us 12 years to establish those trees in a breeding orchard, to make the crosses, produce the seed, get those out to field trials so that we can then measure and analyze and calculate breeding values to make selections. Once that happens, if that's on time in the first 12 years, it then takes at least 2 years to establish an orchard for commercial or large scale seed production. It's going to take about 8 years for that orchard to develop into a large enough or sufficient level of seed production to make it commercial. It then actually takes in our southern pines up 2 years to produce the seed. So it's from the time pollination takes place, it takes 18 months for that seed to mature and develop. And then it takes 1 year in our seedling nurseries to get the seedlings up to size. It's about a 9-month process, but basically it takes a year. So all that to say that is the timeline just to get the product to the customer. And then the customer has a crop rotation of 25 years to grow those trees so that they're large enough to harvest. So to really capture and realize that gain, it might take 50 years or more. So with this in mind, this is where we have to balance how aggressive we're going to be, what kind of chances or risk we can take with technology or with approaches to speed things up. So just thinking about how we operate in forest trees. So moving down the left side here would be our research breeding or germplasm development efforts. And we're currently in the NC State program in particular, we're transitioning from the fourth generation of breeding to the 5th cycle. Those are overlapping a little bit, but these can be an ongoing breeding effort. You can make them discrete cycles, or you can just have a more or less rolling forward breeding effort. So that can be ongoing and never stop. And so you're always improving populations to develop new material. But at least at some point you have to draw from those populations to establish what I'll call, your elite population or elite parents, which is what you're going to deploy. And this is where that idea of gain versus risk comes in again, because we have to characterize those trees. We get an initial breeding value result from our breeding program, but knowing that we're going to produce millions of these to deploy them on the landscape over thousands of hectares or more, we want to solidify that data. So that takes a little time to basically to validate or confirm with additional trials. And this is where, mitigating risk of the unknown. If you're conservative there, you're going to have a larger deployment population. If you're aggressive, you're going to have a smaller, and that does impact, for any given trait, how much gain you can make. And so this is where historically we are probably conservative as an industry. We like to have more trees, we like to have more options, because as we think about that long-term, multiple decades of development and production, something bad is going to happen. So we inherently like to throw in some extra parents in our breeding populations, but that does come at a cost. And so we have to think about what we're giving up when we do that. So really thinking about today, making this presentation, I think this is an ongoing challenge today, is how do we incorporate new technology? What lens do we look through when we look at that technology to think about making gains? And so how can we incorporate new technologies, new methods, look at things differently without setting ourselves up for a disaster down the road? So that's it. All right. So one thing this is already being implemented, but this is accelerating our research breeding cycle to move faster. This could also be used to accelerate commercial seed production, but this is top grafting. Hard to see with the picture, but on the left here you can see that the person here is we're putting grafts we're installing top grafts. This is done again at it might be 15 or 20 meters in height. It might be 25 meters in height, but the crown of the mature tree will stimulate flower production. These are grafts in their first growing season, and quite often you see that they will have flowers 1 year after grafting, which is a great result for us, because if you allow a tree to develop on its own, it may be 7 to 10 years before it's flowering consistently, and so you got to wait a long time to do those pollinations if you're not using top grafting. And then on the right here, you can see we have flowers inside the pollination bag, and that's just 1 or 2 years after breeding in most cases, or after grafting, so we can move forward with the program. Our field trials, we spend a lot of time. Here's just a brief description of what a current experimental design might look like in forest trees. Often a row column design to account for spatial variation. We use single tree plots, so that every tree here inside of a rep is randomized. In the past, we've used block plots where there's a larger number of trees in there, but right now it's very efficient statistically to work with single tree plots. We're constantly looking at this design. How many sites do we need? How many replications in the site? How many families is appropriate to test so you get good variance component estimates? And how to connect the data. So we may work with hundreds of families in the breeding program. We can't test them all in a common garden on one site, and so we have to put them in some type of series structure, make sure the families are connected from one site to the next, so the data can be combined into a large multi-environmental trial data set. This is just an example of what that might look like in the field. So you've got, here's a case where we got 6 replications. This had 170 entries. And you can see from the aerial photo here that nearly complete weed control. The only thing on the site is bare soil and the trees in the trial. So this helps us maintain a good uniform environment, allows the trees to grow, and we can make early assessments on the performance of those trees. So when you're doing that, this is what a progeny test would look like. This trial is starting its second growing season. They're already over a meter tall, which is a very good start. But you can see with weed control and management, there's really no competition to slow the trees down. When we have all that data and we assess the populations, we can start to look at performance. And so here's just a graphic highlighting the potential that you have by selecting and deploying the best genetics. And so at the top here, we have distribution of an index for the full population. And so this goes back to those 4 traits I talked about earlier. So we put those in a ranking index. You can see the weights there where volume growth accounts for half of the equation. The other 3 traits make up the rest. And so then we can look at those best combinations of trait performance. If you select the best half sibs or single, parents, and look at half sib deployment or open pollinated seed, we can see we are amplifying the best individuals from the top end of that population. So we're capturing performance equal to the top of the population. That open pollen, where we don't know what the pollen parent is, it's a wind pollinated species. We don't know the mixture of pollen parents there. That's going to limit our gain, so we can't really go above the distribution. However, if you take the best parents on the top end of that distribution and you make control pollinated or full sip crosses, those full sip families will outperform what you see in the open pollinated families. And so that's one of those opportunities we have to make additional gain. That does constrain the diversity, because you're moving, instead of one mother tree with an unknown number of pollen parents, you're moving to 2 parents in that cross. But you are able to capture considerably more gain in the traits. Another place when we think about gain versus risk, as we establish seed orchards. I mentioned earlier that this may take 8 to 10 years for these orchards to come into seed production, but they tend to last 25 to 30 years. It might not be quite so long if the genetics of those orchards is no longer relevant, but typically they will be used and managed for at least 25 years. So there's considerable effort put in to test those parents, to be sure of what's going in the orchards, and then managing these orchards is a considerable effort. A lot of planning goes in. So here's just a view of those trees being laid out, maybe similar to fruit or nut production. In their initial establishment, often we use complete fertilizers. We'll use irrigation and weed control to make sure these trees get off to a great start. What we're really after is managing the crown of the trees to produce flowers, so that we get good seed production. And so here are just some pictures of what the flowers or strobili look like in loblolly pine. So the female strobili are on the left and the pollen is on the right. So I had some pictures of what the research breeding and pollination might look like. We actually have adapted, as an industry, we've adapted that process over the last 25 to 30 years to do it in a commercial large-scale basis, so that we can deploy the very best genetics in those full sib crosses. And so we actually are able to use controlled pollination to produce seed. It is a very large effort. The last few years, we've installed more than 1 million pollination bags on the trees. So it's a labor-intensive process. We're working at heights of 15 to 25 meters, so we're using lift equipment to do that. But we're able to produce tens of millions, if not hundreds of millions of seed through this process to deploy superior genetic gains on the landscape. So just thinking about all of these things, a couple more pictures just to give you perspective on that effort of how much risk are we willing to take to capture that best gain opportunity. So that's an ongoing discussion, ongoing balance of how many parents to use and how large to take this effort when we think about managing risk. This has been widely accepted in the market. I think we're probably approaching 20 to 25% of all seedlings planted of loblolly pine in the Southeast have been controlled pollinated. That's been growing. The NC State Tree Improvement Program has been tracking that information for the last few years, and so you can see that it has been steadily improving or increasing in scale, particularly the last 10 years. So it's a significant effort, and it's received in the market, and so this is probably something that will stay and continue to grow. Most of our breeding program, operates from 0 to 6 years. So we are, in most cases, measuring progeny tests by year 6, between 4 and 6, making assessments, analyzing data for breeding values, making new selections to go back to our breeding orchards where we generate a new population. And that's for the tree breeder, that's most of our effort stays in that 0 to 6, maybe 0 to 10 year age for trees. But as I mentioned earlier, the actual crop that's produced for the landowner or the farmer is going to be 25 years. And so there have been some previous studies looking at performance over time. There's some long-term studies that basically helped identify the optimum selection age being around age 6 or 8 years. But we do like to see that the genetics we're selecting and the families we're deploying do actually produce what we are projecting them to. So this was a case study. The study site was called Pigeon Pond. We have published this in the past in some white papers, but this I thought would be a good example of what we do see with our genetics. This was basically a yield trial that one of our partner companies installed in 1998. It's got 5 families, 2 that were full sib crosses produced, and then 3 kind of market standard open pollinated families. They were planted in blocks so that we could estimate per acre, per hectare yields with some confidence. It's just a picture of what those trees looked like. So this was, we started measuring at age 12. We were able to take it out to 20 years. But today I've got the results from age 12 from previous analysis. Basically what we saw is, I mentioned the saw timber potential score, that the 2 full sib families that have the AGM codes here, AGM 22, AGM 29, have the largest proportion of high quality stems. So that's these green bars here for the trees. Both of those were at 80% or higher for quality trees. You can see that the other 3 families were lower, between 53 and 66% stem quality. Still not bad. Over half the trees are quality, but not as good as the best genetics here that have been planted. And the purpose of this trial was really to look at this and have confidence that the investment in the pollinations on a large scale were going to deliver value. Calculating the financial returns on these, looking at the overall management and projected yields from these blocks, different genetics, there was a significant improvement. And so particularly with that AGM22 family, more than double what the industry standard families were modeled to produce. So significant value can be observed. You can see it in the field. You can calculate the numbers with the yield and cost and see that it is a good opportunity financially for someone to consider. One of the other questions, and this is just one site, so it's not a definitive answer, but what we did see here that gave us as a company confidence to continue pursuing full SIB family development was the gain differentials that we saw between the families was pretty similar to what was coming out of the genetic trials. And so a lot of times those are questions we get from landowners or stakeholders that we're selecting at young ages. How does that correlate to that final result at harvest? And so in this case, we've got gains. So these are the two full SIB families, and this is just reporting the gain against that industry standard family, AG373. And so these numbers on the right, these are actually the gains from the breeding value calculations from an independent set of trials from this test. So this is the breeding value calculations from the progeny trials, and which pointed to one family being 3% better than that industry standard. The other one is 20% better than the industry standard. The rest of the data points on these lines are calculations at different ages from the yield trial. And so the square in green shows over time, so starting at age 4 up to age 12, we see a change in that best family. It started out a little bit larger, maybe got off to a larger, better start at age 4, but by age 6 to 12, it's really kind of coming in very close to what the progeny test gains showed. So this isn't a guarantee this is going to happen all the time, but we do have a very nice result here with replicated data pointing to the gains out of our progeny tests can be relied on when we're making decisions and looking at what kind of gain we can theoretically capture. And for our landowners, for customers, again, we work with young trees in our breeding program, but being able to demonstrate the value at a tree that's more of a mature size, has real value in the market, is a very valuable data point to have. Even if it's just a case study or one site, one experiment, it's a very valuable data point. Lets people see when those superior genetics are expressed. This is very valuable. I'm going to shift gears and just talk about rust resistance, disease resistance. So I mentioned fusiform rust was a native disease. It's caused by a Crenartium pathogen and is basically present across the native range of loblolly pine. You can see some fairly recent incidence maps on the left here, and heat maps showing large areas that are high risk for disease incidence. Pictures on the right are just maybe some severe examples of what can happen with infection. But basically, if it doesn't kill the tree, which these two are likely to die, it's definitely going to reduce the value of your plantation. So we did several analyses with some genomic resources, did some association testing, and we actually found in some breeding populations several SNPs that were associated with rust resistance. This lined up very well with previous research. There's a good body of research that points to something like 7 to 10 large effect QTLs that are affecting rust resistance in loblolly pine, and we tried to replicate this and had some good results. So we were able to find several SNPs that had statistically significant results. I've got this top left SNP identified, SNP 2374. This is one that was of interest to us. It didn't necessarily stand out in the overall population, but it was a SNP that was informative in a couple of commercial crosses, commercial full SIP families that were of interest to us. And so we decided to explore that a little bit. As we dug into the data, it looked like a more compelling marker to work with. And so we just tried to walk through this process to see if we could validate the initial result. And so this came from some Bayes C analysis. We were exploring a lot of different analytical approaches at the time, and the Bayes results really clarified and highlighted those top SNPs with the probabilities here. So I'm just showing that. There is no order, so these SNPs aren't mapped in any kind of genetic order. These are just numerically numbered. There is not a resource to work with at this point where we can place all of these SNPs on a map or a chromosome. So we don't really know where they were located when we identified them. We just knew they were significant. But this one SNP, 2374, was present in 2 families of interest. And so we do have a laboratory here connected to the U.S. Forest Service that can do controlled inoculations in a greenhouse. So we utilize that resource. We sent seed. So we're retesting the same families where we identified the marker, but it's an independent set of seed. It's not connected to the original trials. And we were able to validate and repeat that result. So the color differences here are different sources of the pathogen. So we had 3 different inocula that we screened against and found very similar results. And so basically, if you have the resistant SNP genotype, on average, you had 11% infection compared to 57% with the susceptible. So it's not complete with one SNP, but we did have a very nice result. The screening center will operate. Basically, they have a typical spore density that they inoculate the trees with, the seedlings with, that we believe for the most part is correlated with field incidence levels. So that's 20,000 spores per mil in the spore density that's applied to the seedlings. We went back and repeated this a couple of years later, and we did it at a higher rate, 2.5 times above the normal. So we had a much higher spore density to screen infection. We didn't know what was going to happen with this, if this would generate 100% infection or something much higher than we wanted to see. But we had a very nice result with the selecting on the single SNP, where against different inocula, we ranged from 10 to 25 to 30% infection in the resistant, where it's 70 to almost 100% infection in the susceptible genotypes. And so we were pretty happy with this result, knowing that it's a single SNP. This isn't going to prevent all infection, but it's certainly a QTL that we want to pay attention to and try to incorporate into our breeding program. We were able to map it. So in this image, we just as a background are showing SNPs or markers that have been mapped previously that are associated with rust resistance. And so here in the middle, SNP 2374 mapped to linkage group 7, which didn't have any other previously known resistant QTLs or SNPs that mapped to that linkage group. Since then, some other groups, and the Tree Improvement Program at NC State, I believe, has identified one on linkage group 7. So there may be more there. That's something we have not pursued further at this point. We did find 2 more markers in some subsequent work that also map to a close location. So there is at least a gene or some suite of genes there that's having an effect. So that's a great opportunity for us to pursue further. Our interest was getting it out onto the landscape and see how it holds up out of a research trial, out of a greenhouse, and get it out into the landscape. And so we put some small plantations out. I think we produced about 200,000 plants. We did this by genotyping individual seedlings. We converted them to hedges and produced rooted cuttings so that we could multiply those resistant genotypes. And so in 2019 and 20, we put several sites out in high hazard areas. You can see on the map there in those orange and red zones. Installed those trials, and we're now in the process of collecting that data to validate. So don't have that summarized today, but we are seeing that the susceptible genotypes that were planted as controls do have rust, do have disease infection, and the resistant materials holding up very well in those plantations. This is probably not commercially viable to bulk up and multiply this way. This is probably, these results will help confirm we need to figure out ways to incorporate these SNPs in a selection approach to incorporate it into the breeding program. But we definitely see value here. So this is the other ideas or other issue when you for us, when we're thinking about gain versus risk, is if we want to mitigate the risk of disease incidents, how much is that worth? And so here we modeled some scenarios of a very good full SIB cross. That product is called MCP, but that's basically, it's a full SIB family versus another full SIB family. If we made a cross that had similar performance, except it had enhanced rust resistance. And so we ran scenarios of looking at that base incidence ranging from 10% to 50% infection. And these aren't out of range for incidents in the field, that you can find. Even the most rust resistant families, there are going to be sites where 30, 40, 50% infection is the real world. But you can see as at 10 and 20% infection, there's really no difference. And so paying for additional rust resistance would not bring anything of value if your disease incidence is low. But you can see as it increases, that enhanced product with additional rust resistance is holding its value, and the regular full SIB family without that added SNP selection we're losing value, and that approaches $200 to $300 in net present value. So a significant difference, and should be a consideration for people going forward, is whether it's rust resistance from a single marker and a marker assisted selection approach, or if it's a tested family that has quantitative rust resistance. You want to make sure, if you're in those high hazard areas, that you're selecting good material, because that's definitely a risk, and you can lose value. This just a snapshot, a couple of pictures here of our rust resistant populations. So everything in these pictures has been genotyped and marker selected. So these are the rooted cuttings that are generated from those seedling hedges, donor plants. And so for us, it was pretty exciting to be able to produce a marker selected product, even at a pilot scale, and be able to deploy it on the landscape. So again, we were able to do that with about over 100,000 plants went out to the field. And then finally, just thinking about that idea of gain versus risk and making genetic gain, we want to have confidence in the trees that we select. And so giving you kind of a overview of a lot of different aspects of the breeding and selection process. Ultimately, it comes down to how good is your data. make those selections to go forward. And so typically in forest trees, well, at least in loblolly pine, we're making backward selections. So we're looking at the progeny test and then going back to the parents of that population to put those into the deployment population. We've got previous experiences where we were aggressive and we made predictions of progeny test seedlings and trees, and we carried those forward rather than making selections to put into a breeding population. We put them directly into a deployment population. And we had some pretty bad experiences where there's a lot of error in the breeding value calculation. Accuracies were low, and we didn't have a lot of confidence. People took that chance, and it didn't work out very well. We've got a lot of different tools now, including clonal replication with rooted cuttings, including genomic prediction and other genomic resources. And so these are opportunities to be more aggressive than we have been in the past. This slide's got a couple of summary graphics of a population that we're working with, exploring some different approaches, and ultimately this graph shows the parental population in green on the far right. This is what we typically see, what we're comfortable with, where accuracy values of our breeding values are approaching 0.9 or higher. Well-tested parents may reach 0.95 to 0.99. So we know we've got a very good data set with typically low error associated with those breeding values. On the other end, in the red bars, this is a range of what accuracies we might see in current progeny tests, where the individual seedlings, those predicted breeding values that we might get out of an animal model are going to range from anywhere from 0.5 to maybe 0.7. And then in the middle is in this blue purple color, these are individuals that have been clonally propagated. So we basically clone the progeny test, replicated it across multiple sites, and that gives us a better estimate of our breeding values, better additive and non-additive variance estimates, and that improves our accuracy. And so we're getting something in the middle. It's not quite as good as the parental accuracies, but still very good. So we're working with an approach here to see if we have enough confidence in those selections to take them straight to a deployment population. That could be done with a combination of genomic analysis, GBLUP, and the clonal replication combined. And so for us, that's an opportunity to press forward in a more aggressive way without compromising on the risk of making a bad selection. I think I talked about all of that. So really just to wrap up, for me, forest trees, working in the breeding program, it's balancing that genetic gain with an acceptable level of risk because of that time scale and the amount of effort and investment it takes to go through the process. We want to make sure we're not messing things up, but we don't want to be too conservative. So that's a constant question of balancing those two. And so we do have demonstrated performance with genetic gain. We know that it's real. And then we must ask ourselves how fast we can move without making mistakes. We know we can incorporate new tools. We want to be aggressive with those to figure out how to use them. And if you look at this timeline of 25 years to go from selection to deployment, we know that a reduction of 3 to 5 years is meaningful for us. And I think with the technologies in place, we can do that. And vegetative propagation and genomics, I think, can play a huge role in improving this and shortening our development timelines and deployment timelines. And I will stop there.
Thank you, Patrick. Thank you. Very, very interesting. Again, post your questions in the chat for the audience. We really have one question, Patrick, is coming from Veronica in Nigeria. It says, I want to know if you have records of G/E performance of your populations outside the U.S.
Measures of G/E outside the U.S. We do have trials in Brazil. I think we see a little more G/E in those populations. So typically G/E, if we're looking at type B genetic correlations in the U.S., they're probably around 0.8. And so we typically don't pay a lot of attention to G/E in the southeastern United States. Those correlations are a little bit lower in Brazil, probably in the 0.6 to 0.7 range. So I think there is a little more interaction there. I think that's the only place I could speak to.
Another question here is the SNPs of interest in this case, are they naturally derived from the loblolly pine? Were they discovered by phenotype or learning from other genetics of interest, assuming no level of interest in GM or gene editing or mutagenesis? Are the results we saw of the individual SNPs or stacks, are the results of the individual SNPs or stacks? The person says I'm familiar with loblolly pine genetics. Does loblolly pine share genomics, any genomics with other pines? Yeah, very interesting seminar.
Try to answer those. The SNP resource where we identified that resistant, the SNP associated with disease resistance was from a public set of SNPs. Those have largely for loblolly pine, those have come from some U.S. government funded grants, and those are naturally occurring sequences in loblolly pine. We have not pursued, related to any of the traits that I talked about today, we have not pursued any biotechnology approaches with modifications of any kind. So everything we're doing here is natural variation that we're capturing with breeding and vegetative propagation.
Good.
Yep.
Patrick, from my own experience of visiting for tree breeding programs, some people may have the impression that, well, a generation interval is 20, 25 years. So what do tree breeders do? They usually ask questions. So around 2 cycles in my professional life, if I'm happy, lucky, but a lot of people don't realize how overlapping generations work. Can you talk a bit about that?
Sure. Yeah, so the breeding populations, one is, we have 5 different regions that we work with. And so those are all on slightly different timelines. So there's no shortage of work to do to get everything done in those populations. But yeah, we may have a population of trees that we work with, and it takes several years to breed. We don't wait to complete all the breeding to move to testing. And so we're constantly breeding, generating material, and getting that out in trials. And so the breeding and the testing, even within a generation or a cycle, is going to overlap. But yes, in some cases, you may only do 1 complete cycle or 2 complete cycles in your career. We're trying to move to where maybe in my career I can get 4 cycles, 5 cycles completed.
That's great. Absolutely. Well, thank you so much, Patrick. Very, very interesting topic. Just as advertisement, if any of the participants is interested in tree breeding, our university is for sure one of the leaders in this area, as Patrick can attest to that. So please contact us. Thank you again for your seminar, Patrick. Thanks for the attending the seminar live. Next week, we're gonna have a presentation by Dr. Ram Nair from the World Vegetable Center, talking about the progress made in mung bean through a network approach. Look forward to having you again at the same time. Have a nice weekend, and let's just stay connected. Thank you, Patrick.
All right. Thank you. Thanks for the opportunity.
Bye.